get a color here. And we have to cover a lot of stuff in a little bit of time. So we're going to get a couple of essential questions, and I'll just go ahead and give you both of them. But we're going to look at what is energy and how does it relate to cells. So what is energy? Okay. And how does it relate to cells? So kind of two things there, looking at energy, looking at its relationship to cells. The other question that's going to come up in the second half is, how do enzymes function? We're going to do a lab with these next time. So we need to know how enzymes function if we're going to do a lab on enzymes. Okay, so those are our two big essential questions for this chapter, or this section anyway. Okay, so cells transform energy as they perform work. Cells are like miniature chemical factories. They thought, this one's going to be a rough one, I can already tell on just me being able to speak and read, apparently. All right, reverse, start over. Cells are miniature chemical factories, so they house thousands of chemical reactions. Some of those reactions release energy, and some of them require it. So we'll get into the specifics of those. Energy is a good thing to define here. You also need this as you move towards physics, so lots of physics concepts here. So energy is the capacity to cause change or perform work. We're going to talk about the performing work one. Okay? So basic forms of energy, kinetic energy is energy of motion. So think kinesiology, it's about motion. Okay? Potential energy is the energy of matter that matter possesses as a result of its location or structure. So it's actually like the potential to do work. So kinetic energy is like moving work. And potential energy is, as it says, kind of the potential to do work. Okay, and that could be based on location, could be based on its structure. Oh, I don't have a good picture for this one. So here, I'll just go with it for you. But it's kind of like being on a roller coaster, as you do the whole climb up the roller coaster, and then you get to the top part. That's where the greatest potential energy is, because at the top of this roller coaster, it's like, I have the potential to have a really great time, or have the potential to have like a terrifying time. If you're me, you guys are young still, and you're not full of terror, so you have lots of potential to do awesome stuff, and then it gets converted to kinetic energy or energy of moving when you go down the roller coaster. Whee! Okay. So that's potential energy. When you're at the top, because of our location of being at the top of the roller coaster, we have the potential to have an awesome time. That's potential energy. Ta-da. All right. Thermal energy is another one. So as so think thermometer type thing. It's kind of a kinetic energy that's actually associated with the random movements of atoms or molecules. So believe it or not, when we're talking about thermal energy or thermal or kind of heat, when we have more heat, okay, we have more movement of atoms. I mean, think about boiling water. You've heated the water up. You get those water molecules moving like crazy. So it's actually associated with movement of atoms. So thermal energy is a transfer from one object to another as heat. Light's also a type of kinetic energy, and it can be harnessed through photosynthesis. That's our next section that we'll talk about. All right, so lots of terms in this one to kind of keep straight. Chemical energy is another one. So this is actually potential energy that's actually available for release in a chemical reaction. So just like being at the top of the roller coaster has a potential to be a good time, if we've got like two atoms that are stuck together, we have the potential to kind of break that bond and release energy. Okay, so there's a potential energy that's stored in bonds. Most important type of energy for living organisms to power the work of the cell is because of chemical energy. Okay. So there's chemical energy stored in foods even. It has the potential to release energy to energize you. That's why we eat. Okay, we're going to come back to this one. This one's not one that you kind of have, but it's a good little video that we can look at. They won't play when I'm doing this screenshot stuff. So, yeah, it goes how it goes. So thermodynamics, thermo, we're talking heat, dynamics is like change, so changing heat stuff, means that we have the study of energy transformations or changes that occur in a collection of matter. So the word system is used for the matter under study. Okay, so surroundings is kind of everything else outside of the system, like the universe. So if we're talking about maybe our own stuff, maybe our body and all the chemical reactions that go on in our body would be the system, and then wherever we kind of are, I don't know, maybe we're at school, I don't know, it's doodles, it's a desk, see my desk, you got a chair, whatever, you get the idea. But that's all the stuff that's going on in there, that's the surrounding. So the system may be the kind of the thing that we're studying, and the surroundings include everything else. Here's your table. Go with it. All right. There are two laws that govern energy transformation. These are definitely some physics concepts, but we need them for biology as well. So the first one is the first law of thermodynamics. Okay, it's energy conservation. So the energy in the universe is constant. It's neither created nor destroyed, is what we say. Energy is neither created nor destroyed. Destroyed. Okay, so we don't create it, we don't destroy it, it's constant. The second law of thermodynamics is that energy conversions increase the disorder in the universe. And so there gets to be more heat loss. We're going to talk about heat loss as it goes on, but when we lose heat, it creates more disorder in the universe. So our entropy is the measure of disorder or randomness. Okay, there's probably your teenagers. I remember when I was a teenager, there was a good lot of entropy in my, my room at home. So it's probably a good lot of entropy in a lot of your rooms. I'm not judging. I'm just saying that's what it is. It's a good way to think about it. All right. So automobile engines and cells actually use the same basic process to make chemical energy of their fuel available for work. So in cars and cells, the waste products are carbon dioxide and water. So you guys should remember this from chemistry of some sort, as that's a combustion reaction. So cells use oxygen in reactions that release energy from fuel molecules. So in cellular respiration, the chemical energy stored in organic molecules is used to produce ATP. If all this stuff doesn't make sense, that's cool. That's what we're going to do next time, so don't worry about it. But here's kind of what we're looking at. So if you know how a car works, you put gas in and it actually takes oxygen from the environment. What happens in order to get kinetic energy, so this chemical energy in gasoline is converted to kinetic energy of movement of the car, we've got to go through that whole combustion reaction, and then we actually end up losing heat. When we lose things as heat, we can't actually get it back to do any kind of work. It's just lost in the system, therefore increasing entropy. Okay, so gasoline, that chemical energy, and oxygen come together to help move the car. You end up with waste products of carbon dioxide and water. Same thing if we have cellular work here. So you take in glucose as kind of your fuel, some sugary item. I mean, you and me both, right? So glucose, then we take in some oxygen from breathing. You're going to undergo some cell work. You actually lose heat energy when your muscles. So as you're moving, kind of like the car does, as you move, you lose some things as heat energy. Okay? And the waste products are carbon dioxide and water. You exhale the carbon dioxide and you get a little water from it. Okay? All that we're going to go into more detail. So if you're a little like, oh, right now, that's okay. Let's just focus on one thing at a time. Okay? So chemical reactions can actually be one of two things. So exergonic reactions or endergonic reactions. Exergonic mean they release, so exit of energy, you can think, for exergonic. For endergonic, it's entering energy, maybe. Okay, so we've got to bring energy in. We require energy. Okay, think that way. So real quick, we'll go through these, but exergonic reactions release energy, the energy exits, okay? So these reactions release the energy in covalent bonds of reactants. So burning wood releases energy, and glucose is heat and light. Cellular respiration, however, we could just burn wood and then we get some good stuff from it, but we actually still need to harvest some of that energy. So cellular respiration is a little bit better than just burning glucose. It actually involves steps, and it releases energy slowly, so that way we can get that energy and produce ATP, which is going to be sort of like a cellular battery. So we're actually able to use some of the food to transfer into an ATP molecule. Instead of just burning it and then more heat that we have, we can actually use that. There's a picture. Went too far. Okay, so what happens? Here's our reactions. That's their potential energy. They'll release energy, and our products have less potential energy. So essentially, we're releasing between this much and this much energy. So that's how we can see what's released. An endergonic reaction, remember, is we need entering, we need sort of it requires energy. It yields products with some rich potential energy. So endergonic reactions start with reactant molecules that contain relatively little potential energy, but end with products that contain more chemical energy. So we get more energy at the end of them. Okay, so if we have reactants that don't have a whole lot of potential energy, but we can add energy to that system, we can produce products that actually have more potential energy, that can have the potential to do more work. So, good example here, photosynthesis will be one of the first ones we talk about, and it's a type of endergonic process. It takes energy-poor reactants like carbon dioxide, not a whole lot in that, and it actually uses the energy of sunlight to help us produce energy-rich sugar molecules. So that's more of the endergonic thing there. Okay. All right. Oh, we can do this real quick. Okay, so a living organism carries out thousands of endergonic and exergonic chemical reactions, all going on together. The total of an organism's chemical reactions is called metabolism. Okay, that's all the exergonic, all the endergonic, all the chemical reactions going on in the body. Not that we think of, like if I move more and I do more stuff, that increases my metabolism. Different definition. When we're talking about the chemistry and all this kind of stuff, we're talking about metabolism as far as all the chemical reactions in a body. A metabolic pathway is actually a series of chemical reactions that either build a complex molecule or break one down. Okay, so either of those. All right, this is probably a good point. We've made it to about 13 minutes. So this is a good point to stop, look at your notes, chunk them. I like to chunk, which is take different sections and put a line in between them if you haven't experienced that yet. That helps me. If it doesn't help you, that's cool. I would write maybe some questions and things out to the side that you can use to study. Maybe start writing a little bit of a summary. Just pause for a few minutes and let your brain rest even. Stretch or get up and jiggle your legs around. Oh, you were there for the sprinter thing. Oh, that was a PSAT thing. Anyways, whatever. Stand up, do something, breathe, and then come back. All right, energy coupling is the next thing that uses energy released from exergonic reactions to drive endergonic reactions, and typically using the energy stored in ATP molecules. So our little, sort of like a AA battery. It's small, but it produces the energy that we need. So ATP stands for adenosine triphosphate. This is a big one, by the way, because we got to know about ATP. Adenosine triphosphate. So it actually looks a lot like, let's see, it's going to look a lot like an RNA. It's going to have a ribose sugar. It's going to have a phosphate group. It has a nitrogenous base that sort of looks like A. Oh, there you go. Adenosine. So, but it's also got a ribose sugar, so it looks more like RNA. But instead of just one phosphate group, by the way, that's our adenosine there, it's going to have 3 phosphates. So I'm just going to draw all over the screen because I can. Boom. 3 phosphate groups, so triphosphate. So that's why it's ATP, adenosine triphosphate, or 1, 2, 3 phosphates. I doodled all over your screen. Alright, so hydrolysis of ATP releases energy by transferring its third phosphate from that ATP to another molecule called phosphorylation. Most cellular work depends on ATP energizing molecules by phosphorylating them. But just like attaching a phosphate group, boop, sticking it on there. So here's ATP, here's a good picture of it. We're going to make some ATP while we're talking. I'm undergoing cellular respiration, this will make sense later, but I'm going to make some ATP while we do this. I'm going to have a snack. I'm eating some of those Twizzlers we have. Some spindle fibers are getting eaten today. And they're delicious. Alright. Adenosine triphosphate looks like this. Adenosine with 3 phosphates. Alright. We can lose between the second and third phosphate group right here as the best potential energy. Kind of like hydrolysis, we had a dehydration synthesis and hydrolysis. Hydrolysis, which is adding a water and splitting. We can do the same thing with ATP. We can add in water and split between the second and third, and it releases energy into the environment. That's how ATP kind of works like a battery. All right, there are 3 main types of cellular work: chemical, mechanical, and transport. ATP can do all of those. We won't really get into it a whole lot. We'll use more ATP a little bit later, but just to give you an idea is that it can do chemical work, so it can sort of like be a catalyst, sort of like what we'll talk about, catalyst, sort of like what we'll talk with enzymes, but it does a little bit different function, but it can sort of make it happen a little bit faster. It can be used for transport work, so ATP is used like what we remember for active transport. That should be what we know already. And then mechanical work, so it actually literally makes all of your muscles move. And that's that. Cells use and regenerate ATP continuously, all the time. In the ATP cycle, energy is released in an exergonic reaction, like the breakdown of glucose, during cellular respiration, which is going to happen right now, so I'm eating, and is used in an endergonic reaction to generate ATP from ADP. So endergonic, so that takes in energy to make the ATP. So sort of what it would look like is if we have ATP and we can have hydrolysis, which is exergonic, it's releasing energy to do some cellular work. If we need to pop the phosphate back on, sort of like recharge that battery, it's like a rechargeable battery, those old things, okay, we can put the phosphate back on by getting energy from a different system, which we will, and then we get our ATP back. All right. This would be a good place to pause, so take a break, review your notes, make sure you got it, and then we're going to talk about how enzymes function. All right, enzymes. So although biological molecules possess a lot of potential energy, it's not released spontaneously. Okay, so an energy barrier must be overcome before a chemical reaction can begin. I told you when we started this was going to be a rough one for talking. And it is. So this energy is called the activation energy, which is pretty descriptive. It's the energy that's required to activate a particular reaction. I mean, it makes sense. Okay? It activates reactants. So we can think of activation energy as the amount of energy needed for a reactant molecule to move sort of uphill to a higher energy but unstable state. So the downhill part of the reaction can begin. Kind of like how much work do we need to put in to get to the top of that roller coaster where things are exciting? So remember that roller coaster? It's like, woo, there's going to be a lot of great stuff here, but it still requires some energy to haul that thing all the way up to the top of the roller coaster. Same thing. So one way to speed up the reaction is to add heat, which agitates atoms so that bonds break more easily and reactions can proceed, but too much heat can actually kill a cell. We don't like it. So we have enzymes. Enzyme function as biological catalyst. Catalyst just sort of means it's speeding up reaction rates. It's just making things happen faster. Okay, it increases the rate of reaction without being consumed by the reaction, so we can actually reuse it. Okay, they're usually proteins, although sometimes we have some RNA molecules that can function as enzymes, but for the most part, we're just going to say that they're proteins. Enzymes speed up a reaction by lowering that energy of activation. All right, that's what it does. Okay, and we're going to look at this video a little bit later too, because there's always these nice animations that help us out here. So here's sort of the activation barrier. So we need a certain amount of energy for those reactants to make it go. And that's where it would be without the enzyme. However, if we have an enzyme in place, notice how much lower this barrier is, because we don't have to do a whole lot to get to the bottom here. All right. Same thing you can sort of see here is that the energy here is that if it's without the enzyme, it's the black line. We have a lot of energy that we have to kind of apply to the system before we can make the reaction go. And the enzyme one, which I'm kind of dotting here, notice it's a whole heck of a lot lower. So we need a lot less energy to make the same reaction and get the same products. Okay, so that's the benefit of an enzyme. An enzyme is really selective in the reaction it catalyzes. It has a shape that determines the enzyme specificity, so they're really specific. Okay, the specific reactant that an enzyme acts on is called the enzyme substrate. The substrate fits into a region of the enzyme called the active site. We'll look at these, but a good idea is to have a little kind of doodle of these things when we get to it. Okay. Enzymes are specific because only specific substrate molecules can fit into their active site. So let's get a figure. Okay, so here's our enzyme, and we'll use sucrase. If it's got ends in ASE, we're going to know they're an enzyme. Okay, so ends in ASE, it's an enzyme. Here's our enzyme, and here's the active site. It's right along here. It just means this is where the substrate, the thing it's going to work on, is going to land. Okay. So we can add the substrate, our sucrose little molecule here, and it binds. Notice the active site is here, so it fits in snugly. We could say it's an induced fit. It just like kind of hugs the little sucrose, the little substrate. All right, then we have some conversion go on. Whatever it is, it makes it happen, but this particular time, it broke the bond between those two, so the substrate is converted to its products. Okay, and then those products are released. So we have fructose and glucose, and they're released out into the environment. And then we have an enzyme useful again with a new active site available for a new substrate, the thing it's going to work on. Okay, so for every enzyme, there are optimal conditions under which it's most effective, which we'll explore in our next kind of labs. So temperature can affect molecular motion. An enzyme's optimal temperature produces the highest rate of contact between the reactants and the enzyme's active site. So most human enzyme works between 35 and 40 degrees Celsius. Think about it, that's around 98.5 degrees Fahrenheit. Oh, why would an enzyme in the body work best at 98.5 degrees? Well, that's because that's what your body is doing. So human enzymes work best at body temp. That should make sense, right? So the optimal pH for most enzymes is near neutrality. They like somewhere around 7. Makes them happy. All right, many enzymes require non-protein helpers called cofactors. These guys help bind to the active site and function in catalysis or catalyzing things. Some cofactors may be zinc, iron, copper, stuff like that. Occasionally, we'll have some other molecules like vitamins that are called coenzymes, and these are organic. We'll have a particular one come up later called acetyl coenzyme A that will be really important for cellular respiration. A chemical that interferes with an enzyme's activity is called an inhibitor and inhibits it from working. So a competitive inhibitor would block the substrate from entering the active site. Right? Let's see if I have a picture real quick. I don't. We'll go back to it. But think about that little sucrase. I'm going to draw my best doodle here. So if this is our sucrase from earlier, if I had a competitive inhibitor, maybe I'd have something that's kind of this shaped that fits in here, and it says no. So this little sucrase dude comes along and it's like, well, I want to sit there. And it's like, no, just no. Okay, so that's a competitive inhibitor. I feel like there should be a meme for that. That would be a good one on that one. Non-competitive inhibitors bind to the enzyme somewhere other than the active site, which actually changes the shape of the active site and prevents it from binding. So if we have the same sort of weird sucrase here, I don't know if I'm drawing it right anymore, but you get the idea. If I have another thing, maybe it sits over here for non-competitive, it would change the shape of this thing. So maybe it's like, oh, no way. Can't let anything in here. So that poor little sucrose comes around and it's like, well, I'm just trying to fit there, but you know, it can't. Okay, so it's a non-competitive inhibitor because I have some other site than the active site is blocking or preventing this from happening. All right, so here's a little bit better. Normal is here's our enzyme and our substrate at the active site. If I have a competitive inhibitor, here it is, and it's going no. Okay, here's this little non-competitive inhibitor, and it sits over here, and still its basic thing is like no. Like not happening. So both of these cases, the substrate can't bind. And sometimes we need that. Okay, this will be kind of where we're close to the end here, but enzyme inhibitors are important for regulating cell metabolism. So in some reactions, the product can actually act as an inhibitor for one of the enzymes in the pathway. This is what we call feedback inhibition. It provides feedback to the original kind of reaction. So let's say we have starting molecule A, because oftentimes it works like this, where we need an enzyme to produce a reaction to get a product, and an enzyme to produce a reaction to get a product, enzyme to a reaction to a product. At some point, this particular product can actually come back to enzyme 1 and inhibit it so it doesn't work and say, you know, we have enough of this particular product that we don't need this one to work anymore. Like, we're good here. I don't need this. So some of this would come back here and stop the rest of this from continuing to produce too much. Because too much for cells can often be just as bad. So we don't want that. So when we produce enough, it comes back to the first enzyme and says, no, 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 we're good here. Okay. All right. Last connection here is that many beneficial drugs act as enzyme inhibitors, including ibuprofen, which inhibits an enzyme involved in the production of prostaglandins. These things that increase sensory pain and inflammation. So this is how it helps with pain and, you know, swelling. Some blood pressure medicines, antidepressants, some antibiotics, and some protease inhibitors that are used to fight HIV, all are enzyme inhibitors. So it's an important role that we need for scientific study. Okay. Enzyme inhibitors have also been developed for pesticides and as deadly poisons for chemical warfare. So that's where kind of the scary parts get, is that it can be used for non-scientific purposes, which is always a little alarming. All right, so we've covered those things quickly. So take your time on it, get the definitions written down, and then when we're ready to get back to it, we'll go through some of the definitions and then we'll go through the lab materials together to make sure that we have an idea of what's going on with proteins and enzymes and all that good stuff. But in the meantime, if you have any questions at all or don't understand the material, shoot me an email and we'll try to make sure that it makes sense.